Welcome to EdexcelBiology.com by Tashmika Rajakaruna. This is video 2 in the Chemistry for Biologists series. In this video, we'll be covering carbohydrates, their structure, types, and biological importance. Organic compounds and biological molecules. Biological molecules are essential for the structure and function of all living organisms. Most of these molecules are organic compounds, meaning they contain carbon atoms. What are organic compounds? Organic compounds are molecules that always contain carbon, often contain hydrogen and oxygen, sometimes contain nitrogen, sulfur, or phosphorus. These elements combine in various ways to form the complex molecules that make up cells and tissues. Carbon atoms can form 4 covalent bonds, allowing them to bond with many other atoms, including other carbon atoms. This makes it possible to build long chains, branched structures, and ring-shaped molecules. Carbon's versatility allows the formation of large molecules called macromolecules. Monomers and polymers. Many biological molecules are polymers, made by linking smaller units called monomers. For example, sugars, monomers, can join together to form complex carbohydrates, polymers. Carbohydrates. Carbohydrates are vital for all living organisms. In humans and animals, they act as a primary source of energy. In fact, the glucose you get from carbs fuels cellular respiration, the process that releases energy. They're also important in food globally. Rice, potatoes, bread, and sugar all contain carbs. In plants, fungi, and bacteria, carbohydrates play structural roles, such as building cell walls. Carbohydrates are made up of carbon, hydrogen, and oxygen, often in a ratio of 1:2 to 1. There are 3 main types, depending on the number of sugar units. Monosaccharides, single sugar units example. Glucose, disaccharides, 2 sugar units linked, example sucrose. Polysaccharides, long chains of sugar units, example starch and cellulose. Monosaccharides are the most basic units of carbohydrates. General formula is CH2ON, which means for every carbon atom, there are 2 hydrogens and 1 oxygen. They're small, soluble in water, and usually taste sweet. Monosaccharides are grouped based on how many carbon atoms they have. Triose has 3 carbon atoms, key in mitochondria during respiration, as intermediates. Pentose has 5 carbon atoms, found in DNA and RNA. Examples: ribose, RNA, and deoxyribose, DNA. Hexose has 6 carbon atoms, most common: glucose, fructose, and galactose, involved in energy release and transport in cells. Glucose exists in 2 isomeric forms, called alpha-glucose and beta-glucose. These are both hexose sugars, meaning they have the same chemical formula and the same atoms, but the arrangement of atoms differs slightly. In alpha-glucose, the OH group on carbon 1 is below the ring. In beta-glucose, the OH group on carbon 1 is above the ring. Even though this is a small difference, it has a big effect on how glucose molecules link together to form polymers. Why does it matter? Alpha-glucose molecules join via 1,4 glycosidic bonds to form starch, amylose, or glycogen. These are used for energy storage. Beta-glucose molecules link differently, forming cellulose, a polymer with straight, strong chains used for structural support in plant cell walls. So, even though both are glucose molecules, their isomeric forms lead to the creation of very different biological molecules with different functions. The bond formed between the 2 monosaccharides is called a glycosidic bond, a type of covalent bond. How is a glycosidic bond formed? It forms through a condensation reaction. One monosaccharide provides a OH group from carbon 1. The other monosaccharide provides a hydrogen, H, from one of its OH groups, commonly carbon 4 or carbon 6. A molecule of water is removed. The oxygen atom left behind links the 2 sugar rings together. Naming glycosidic bonds. We use numbers to show which carbon atoms are involved. 1,4 glycosidic bond. Carbon 1 of the first sugar bonds with carbon 4 of the second. Common in maltose and amylose, part of starch. 1,6 glycosidic bond. Carbon 1 of the first sugar bonds with carbon 6 of the second. Found in branched polysaccharides like Amylopectin and Glycogen. Glycosidic bonds can be broken by hydrolysis, the reverse of condensation. A water molecule is added. An enzyme, example maltase, sucrase, or lactase, catalyzes the reaction. The bond breaks, releasing 2 monosaccharide units. Disaccharides, double sugars. Disaccharides are known as double sugars. They are made when two monosaccharides, single sugar units, join together. This reaction is called a condensation reaction. It forms a new bond and releases a molecule of water. Disaccharides are also soluble in water and have a sweet taste. Here are three well-known disaccharides and the monosaccharides that form them. Sucrose. This is the common sugar we use every day. It's mainly obtained from sugarcane and sugar beet. Sucrose is made of two monosaccharides, glucose and fructose. Lactose. This is the main sugar found in milk. It is formed by the combination of glucose and galactose. Lactose is especially important in the diets of infants and young mammals. Maltose. This sugar is commonly found in germinating barley. Maltose is made up of two glucose molecules bonded together. It's an important sugar in the production of malt and beer. Our bodies break disaccharides down into monosaccharides using enzymes like sucrase, for sucrose, lactase, for lactose, maltase, for maltose. These enzymes work by catalyzing hydrolysis reactions. That means they help break down disaccharides by adding a molecule of water. This water helps break the glycosidic bond that holds the two monosaccharides together. Let's look at a few examples. Maltose is broken down into two glucose molecules when water is added. Sucrose splits into glucose and fructose. Lactose breaks down into glucose and galactose. This process is essential because only monosaccharides can be absorbed into the bloodstream and used by our bodies for energy. Polysaccharides, the complex carbohydrates. Polysaccharides are the largest and most complex carbohydrates. They're made when many monosaccharide units, usually glucose, are joined together by condensation reactions. Each of these reactions forms a glycosidic bond and releases a water molecule. When have 3 to 10 sugar units, we call the molecule an oligosaccharide. When have 11 or more, it's a polysaccharide. Polysaccharides do not taste sweet, unlike simple sugars, but they serve vital biological functions. Key properties: insoluble in water, won't dissolve easily. Compact, fit large energy stores into small spaces. Chemically inactive, do not interfere with other cell reactions. No osmotic effect, do not affect water potential, so they won't draw in water. This makes them ideal for energy storage. When energy is needed, hydrolysis, the reverse of condensation, breaks the glycosidic bonds by adding water, releasing glucose. This process happens in the gut, during digestion, in liver and muscle cells, when stored carbs are broken down for cellular respiration. Starch, energy storage in plants. Starch is the main storage carbohydrate in plants. After photosynthesis, the glucose produced is quickly converted into starch because it's insoluble, won't affect water potential, it's compact, good for storage, it's easily broken down when needed. Starch is made of two polymers of alpha-glucose. 1. Amylose, a long, unbranched chain of alpha-glucose, joined by alpha-1, 4 glycosidic bonds. As the chain grows, it spirals into a helix. This makes it very compact. Releases glucose slowly and steadily. 2. Amylopectin, a branched chain of alpha-glucose. Mostly alpha-1, 4 bonds, but with alpha-1, 6 glycosidic bonds at branch points. It has many terminal ends, so enzymes can break it down quickly. Glucose is released rapidly when needed. Amylopectin gives a quick energy release. Amylopectin is highly branched. It contains alpha-1, 4 glycosidic bonds along the chain and alpha-1, 6 glycosidic bonds at branch points. These branches create many ends, where enzymes, like amylase, can attach and start hydrolyzing glucose units. More ends creates more access points for enzymes. This allows multiple glucose molecules to be released at the same time. So, energy is made available quickly, ideal during bursts of activity. Amylose gives a slow, sustained energy supply. Amylose is unbranched, long, helical chain. It contains only alpha-1, 4 glycosidic bonds. Forms a spiral shape, which is compact but gives only 2 ends, one at each side of the chain. Fewer ends equals limited enzyme access. Glucose is released 1 unit at a time from the ends. This results in slower, steady energy release over time. Glycogen, energy storage in animals. Glycogen is the animal equivalent of starch and is the main carbohydrate storage molecule in animals and fungi. It is sometimes called animal starch. It is stored in liver and muscle cells, where it's broken down into glucose when needed. Glycogen is chemically very similar to amylopectin but with a more highly branched structure. Why glycogen is ideal: very compact, stores a lot in a small space. Insoluble, no effect on osmosis. Highly branched, fast energy release. Found in tissues that require quick energy, like muscles. Structure. Made of alpha-glucose units. Contains alpha-1, 4 glycosidic bonds along the chains. Has many alpha-1, 6 glycosidic bonds, creating frequent side branches. These branches mean more terminal glucose units. Faster breakdown by enzymes. Rapid glucose release when animals need energy quickly, example, during exercise or fight or flight responses. Cellulose. Monomer unit. Beta-glucose, not alpha-glucose like starch, glycogen. The chemical formula is the same, C6H12O6, but the orientation of the OH group on carbon 1 is above the plane in beta-glucose. Bonding. Joined by beta-1, 4 glycosidic bonds. To form this bond, every second beta-glucose must rotate 180 degrees, so the O groups align. This rotation gives cellulose a straight, uncoiled structure. Chains and fibrils. Individual cellulose chains are straight and unbranched. Many of these chains lie parallel to each other and are held together by hydrogen bonds. These form microfibrils, bundles of cellulose chains. Microfibrils group into fibers, extremely strong and rigid. Why is cellulose so strong? Hydrogen bonding. While each bond is weak individually, thousands together provide incredible tensile strength. Straight chains. Unlike coiled starch or branched glycogen, cellulose chains don't spiral. They stack neatly and support each other. Microfibrils. These provide high mechanical strength and resistance to stretching, essential for cell wall integrity. That's the end of our video on carbohydrates. Let's meet in the next video, where we'll be learning all about lipids. Don't forget to subscribe to be notified when new videos are uploaded. Thanks for watching.